Chromosome number 1, we have one from mom and one from dad. Okay? Chromosome number 2, we have one from mom, one from dad. Okay? Now that's just like the karyotypes. Do you remember doing the karyotypes? Right? So how many chromosomes number 1s do we have? Two. How many chromosome number 2s do we have? So this is, if this were just in a cell, right? That's called the diploid number. Diploid. 2 sets of chromosomes, right? One set from mom, one set from dad. Are we good there? Okay. So this is the cell. They're the chromosomes, and it's an interface. Okay. And there's the nucleus around the cell, or around the chromosomes. And during interphase, the chromosomes replicate. Hey, which part of the interphase does that happen in? Synthesis, S phase. Very good. And then but they're not all wound up like that. They're very loose, uncoiled, so that the DNA can replicate. Okay? So there's your chromosomes replicated inside the nucleus. Okay?
So there's a nucleus in mitosis and meiosis?
Correct.
No. Okay.
Correct. Now the nucleus is going to start to dissolve. It all dissolves away. And you have, let's get some centrioles here. We gotta find some, here's the centriole. I found a centriole. Wait a minute. Centrioles. Centrioles are together, and they're going to go to opposite sides of the cell. Okay? And the area in which the centrioles are found? Centrosome. So from the centrioles out come these tiny little fibers, microtubules. And these things are going to connect to these little grappling hooks on here called the kinetochores. And it's going to kind of bat these things back and forth. Now here I have the centromeres, centrioles at opposite sides of the cell. Spindle fibers setting up, nuclear membrane gone, prometaphase. Are we okay? Okay. Now what are we doing? Are we doing mitosis or meiosis here so far? Mitosis. So what happens here is you're going to have these chromosomes, right, lining up. They get tugged and pulled by the spindle fibers until they line up in the middle. Middle between the centrioles. This is metaphase. And then those spindle fibers go pulling the chromosomes away. Hey, can I call these chromosomes chromatids?
Oh, yeah. No, you can't because they're apart.
I can call them both, actually, at this point, okay?
They're copies.
Yeah.
Okay.
Okay? So it would be like saying they're sisters. Okay? Okie doke? So they go to opposite sides of the cell, centrioles there. Okay, and now we have 2 identical cells. Right? Okay, so if I look back here, I have one chromosome 1, one chromosome 1, one chromosome 2, one chromosome 2. Haploid or diploid?
Haploid.
Diploid. 2 copies of each chromosome. Hey, these 2 chromosomes, what are they? What's another term that you can use? Call them. They have the same information. Homologous. Same information. Okay? Same type of information. Now, one might say blue eyes and the other might say brown eyes. Okay, so they might have different alleles, different choices, but they both have eye color, chin shape, elbows, whatever. Okay? Are we okay with mitosis? Okay, so we're back to the beginning. Diploid cell, centrioles all together, right? And so we're in interphase, right? Interphase. And in interphase, we have the S phase. And S phase, chromosomes replicate. Dunkada, 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 dunkada. Oh. Dunkada. Okay? Nucleus is still there. Okay? And nucleus starts to dissolve. Oh, simultaneously, those centrioles have to move. Okay? Centrioles are going here, setting up spindle fibers, and they hook these bad boys. Wait a minute, that's wrong. No, not for meiosis. Right? These are tetrads. Okay? Formation of homologous chromosomes and their pairs, right? So we have 4 chromosomes that all have the potential of having eye color, right? Now, are they all the same type of eye color? Nah. You have blue, brown. Maybe you do have all brown. Okay? And, yep, go ahead.
Is this whole thing a tetrad?
It is.
Just 2 tetrads? Oh.
Tetrad. Did you ever play the game Tetris? How many blocks are always falling? Always 4 blocks. Go home and play Tetris for a little bit, and you will think biology, tetrads. Okay, it's always 4 blocks falling.
Why are they like?
No, they're like this. Okay, but that's because these are kind of rigid. If in real life we were looking at chromosomes, this would probably be more like them, right? Where they're more flexible. Right? Here's Here's a chromosome. Here's another chromosome. Mom, dad, right? S phase, synthesis, and then they come together, right, in prophase, and metaphase, 1. But when they go like this, look at there's parts where these two chromosomes cross over. And what can happen is that they can detach and actually exchange information, right? So crossing over can happen in prophase 1 up until metaphase 1, whenever they're in a big old group like that. Maybe here they cross over. Okay? Crossing over.
And when is crossing over? Like what phase is that on?
Whenever these chromosomes are in a group of 4. So when would they be in a group of 4? How about metaphase? And when they're the chromosome, right? Centrioles back there, prophase. Now we call this prophase 1 and metaphase 1. Nowhere else do you ever see 4 chromosomes coming together, 4 chromatids. Okay? So here, prophase, metaphase, right? Now anaphase is going to happen, right? And the homologous chromosomes separate, okay? And they go to their own cell, okay? So now over here with Morgan, right? Actually, in between here, you're going to have chromosomes that actually replicate their centrioles again. Zoop, right? And now look at, hey, copy and original separated. What's this look like?
Mitosis.
Mitosis. Looks just like mitosis. So how do I know that this is meiosis 2? Metaphase 2. There are 2 chromosomes. If that was metaphase 1, what would it look like? Metaphase, I would have 4 of these bad boys. Right?
So it's going to make haploid cells.
Correct.
So that's the only way that they're different, is that they're haploid?
Right. But now look at, wait a minute. If I look at these, I have big blue, little red. Big blue, little red. So let's go back to these. Red and yellow separate, right? And then red and red separate. Really? Are they the same chromosomes? They're a tad bit different. How come they're different? Crossing over happened. Okay, so as much as right now it looks like those chromosomes are the same, they're really not. Okay? And crossing over is not going to happen the same way all the time. Now, when I look at my chromosome, right, I have a gene here, and I have a gene way up here. What's the likelihood that crossing over can happen between here and here compared to here and here? Why?
Because they could be split.
Right, they're farther apart, and there are more places that they can split. Right? So if we're looking at gene mapping, we can say, hey, if recombination happens between here and here, a lot, and recombination happened between here and here, not so much, we say, oh, then these 2 that are not so much have to be close, whereas the ones that happen a lot have to be further away. The more different, the further away. The more similar, the more close. Does that make sense?
Mm-hmm.
Okay, so we're okay with meiosis? Yeah. Meiosis 1, what can you tell me about meiosis 1? You have tetrads, and you have a whole chunky amount of chromosomes going on, right? Right? So here you have 2 chromosomes coming away from 2 chromosomes in anaphase 1, right? Anaphase 2, what's that look like? Mitosis. Is that okay? Okay, so let's go to Mendel's law, right? Mendel's law. Hey, here, when they sorted, they came apart, right? I had dad's chromosome going to one side, mom's chromosome going to the other. Mom's going to this side, dad's going to this side. They independently sort. Chromosome 1, just because dad's went off to the right here, doesn't mean dad's second chromosome has to go off to the right. They can sort independently of each other. Do you see what I mean?
So are like these 2 always together and those 2 always together?
Yeah, because these are copies. They're well, always until they hit meiosis 2, right? And then they separate. And they look like they're the same, but again, they're not going to be because of crossing over. Okay? And crossing over is not always going to happen the same way. If I gave every single one of us a pack of these little guys and said, okay, plop them on your table and see where crossing over happens, do you think we're going to have the same patterns in all of us? No. Okay? Is this okay? Okay, so we're okay with independent assortment. That just means that the different number of chromosomes will separate independent of each other, right? Chromosome number 1 separates independently of what chromosome number 2 is doing. Chromosome number 3, the next one, is going to be independent of what 1 and 2 do. We have 23 pairs. Holy moly. Okay? How about in you? Okay, let's go back to our diploid cell. 2 copies of each chromosome, right? Okay, so this is what's in every cell of our body with the exception of egg and sperm, right? Okay, so that's our diploid chromosome number. Here, let's say that on here is eye color. And look, eye color. This one has blue eyes, and this one has brown eyes. Okay, so if we look at that person, those are the genes on their chromosomes. If we look at this person, what are they going to look like? What's their phenotype? Brown eyes. Now, just because somebody's brown-eyed and good-looking, right? Does that mean that their children will always be brown-eyed? No, because when this goes through meiosis, right? I can actually make a sex cell that has blue eyes and a sex cell that has brown eyes. So, right, and they separate. That's meiosis. So these traits separate. Mendel's law of segregation or separation. They segregate, they separate. So that just because they come together and in you they're brown eyes doesn't mean that that's the way it always has to be. Those 2 traits can then separate. So variation. Can you see where variation with independent assortment can occur? A brown-eyed person can have a blue-eyed child, right? Variation with crossing over. Are we good? And variation with independent assortment. Just because dad's chromosomes came in a package deal, and mom's chromosomes came in a package deal, they get meshed together, and at some point meiosis happens, and they separate independent of each other. The different chromosomes separate independently of each other. Good? Okay. Ta-da. You're very welcome. Wasn't this almost fun? Look at that. A gene becomes a centriole. Oh, these things can just become anything.
I like seeing it like that.
The fun, I know, science is